Hi, I'm Rich with insideHPC. We're here at the Intel HPC Developer Conference, and I'm here with Erik from Stockholm University. Erik, welcome to the Intel conference. I'd like to start with a question about your work and the challenges you're facing at Stockholm.
Sure, we've done HPC for a long time, in particular in molecular dynamics and simulations, but the part that's new for us right now is that we've gone heavily into cryo-electron microscopy, which is incidentally also the topic of this year's Nobel Prize for Chemistry.
So, I've read a lot about Cryo-EM. How is Intel helping you with that?
So the challenge with Cryo-EM is that it's an experimental technique, but it's an experimental technique that generates tons of data, hundreds of gigabytes. So to make sense of this and actually be able to get a structure of the proteins in our bodies, we need to use immense amount of computation to really sort out what the structure looks like on a nanometer level. And this is so much that normally you had to wait weeks or months for the supercomputer to do this, and what Intel has helped us to do is accelerate this so we can get it done in, say, a day or so, or even on my laptop sometimes.
Well, this is interesting because this is essentially like a computational microscope, isn't it? Isn't that what you're doing?
It is in a way, but I think the beautiful thing is it's not really a computational microscope. It's a real microscope, but the data we get out from it, we have to use computers to actually interpret. So it's kind of blurring the whole line between what is an experiment versus what's a computation.
Okay, Erik, so where is this going? What excites you about the future of Cryo-EM?
I think what excites us about the future in general of computers and life science is really that it's certainly nice to get a factor 2 or 5 speed up of a program, right? But by the time you're accelerating things by an order of magnitude of more, the computer starts to change how we do the science. Suddenly you can ask questions that were not possible to ask before, that we can get more biological information from the problem, not just doing it faster. And I think that's what we're starting to see happening in Cryo-EM now, that we can get more data because of the computers, and it helps us understand the biology better, not just faster.